Todd McDevitt, let's see, bachelor's degree from Duke University. Was it a double major, biomedical engineering and electrical engineering? Occasionally he sees an electron, but went on to Seattle to get his PhD at the University of Washington, working with Pat Staten and Alan Hoffman, and then did a postdoc with Chuck Murray, who's on the clinical side, but an academic clinician doing serious research, and actually, it's the Stem Cell Institute here now.
Yep.
So, and then came here in 2004, and his academic appointments in biomedical engineering, but very much a member of IDB. And I'm thinking, Todd, that maybe we ought to get going, even though there are a few people still straggling in.
That's fine with me.
So, and for those that are straggling in, you may have to sit at the front. There are plenty of chairs up front. So.
Thanks, Bob. And there are plenty of seats up here. So, it's a real pleasure for me to have a chance to give this lecture here. For one, this is the type of thing that, and I think you've heard this from many of the previous speakers who have had this opportunity to give a Breakfast Club seminar as well. We often travel and give these types of talks, so we get to explain the story to a lot of external folks. It's rare that we get a chance, perhaps, to go at length. I won't go too much at length, but I'll go at some length about what our labs do, and in particular, try and highlight some of the different aspects today of what my lab in particular is focusing on, which I've identified or titled as engineering stem cell technologies. So let me give you a little sense of what do I mean by that and where I think this fits within the context of the field. I'm sure that many of you are familiar at least with the term and at least the idea of what is a stem cell. So I'm not going to spend a lot of time on that. I'm also, anyone who was fortunate enough to hear Fiona Watt speak last week, I think gave, as I told her afterwards, perhaps one of the most concise, most efficient overviews of the entire field that I have yet to see in terms of what are the major issues and all the different types of research that people are approaching. And so within that landscape, and much of what Fiona spoke about was the amazing discoveries that we've seen in cell biology and the history at which this has occurred over the last really 40 years, but in particular in the last 10 to 20 years where we've seen it really explode. And what that has really involved has been really the ability to think about new ways to derive, identify, and obtain these cells, which has allowed us then to be able to study properties of these and what are the functional characteristics of stem cells. Not only their ability to self-renew and propagate and form more themselves, but also a key feature which makes them very interesting to a number of researchers, in particular our lab, is the ability of these cells to differentiate. And when you start to think about the potential to transform a cell into a variety of different cell types, then it opens up a plethora of different types of applications that you can begin to envision as well. Any of these can be on the diagnostic side, new in vitro platforms for both drug discovery as well as new types of perhaps as yet unidentified types of diagnostic tools, simply due to the fact that we have not yet had some of these cell sources before. So it's new types of assays in that area. And one of the areas which I'll talk about a bit about as well with regard to is with regenerative medicine and the idea of actually being able to use these cells and really start to think about mechanistically how do we use these cells appropriately to actually begin to move towards repairing tissues, which is oftentimes one of the very high-profile advertised goals of this field. So why aren't we doing this yet? That should be the question that maybe some of you are asking, and certainly the question that I was asking as I started to enter into this field. I've heard about this, and I've seen some of the science, and there's great science going on. So why are we not seeing some of the applications develop? What is the chasm that perhaps exists between these two fields or these two aspects of the broader field? And this is then where I started to think about, well, the traditional role of engineering in particular is to basically transform basic scientific discoveries into actually practical and usable and valid applications. And so I think that that's where our lab particularly interfaces particularly well with this, and that is thinking about how can we actually engineer stem cells or develop the engineering systems that are necessary to really be able to transform some of this science into application. And I think this encompasses three main things, and three of the things that my lab is focused around. One is how do we properly control the cells, particularly through control of extracellular environments. How do we do this in a way that's not just practical at the lab bench, that we can have some nice papers, have some systems that work, but think about from the very beginning, how are we going to actually develop systems that will will enable us to be able to scale these up as we move forwards towards some of the biotechnology and the bioprocessing of the cells. And also then, what is the final product going to look like? It's one thing to be able to produce a plate of cells, but a plate of cells is not what a clinician is looking for. It's not necessarily what a patient is looking for. And so we have to think as well about the whole process all the way through, and then start from the beginning with that kind of end goal in mind. And ultimately then, this is how at least I envision that we can perhaps actually translate what at the moment is much hyped and much discussed potential of stem cells into something that will actually be practically useful. So in the current landscape of the field, and this is my brief one-slide summary to try and capture what the main aspects of the research are, there are a number of different types of stem cells, in particular pluripotent stem cells, which my lab is focused on, and there are a number of other progenitor cell types that can be derived either from this cell type or from other types of other cell or tissue sources. These cells can then go on to differentiate into this vast array of cartoony or these cell types depict in this cartoonish fashion here, which could then be relevant and applicable to a variety of different types of, as I said, applications. Some of these could be disease repair models. Others could be new platforms for drug discovery. What has created a lot of excitement in the field, and I've depicted in a manner with the arrows moving in an opposite fashion, is the idea that we can actually begin to take cells that exist in this fully differentiated or somatic state and actually begin to reprogram these. And this has really only been achieved in large part due to the identification of the factors that exist within the cells at this early primitive state, as well as some of the cells in this intermediate state. And so by understanding what was going on here, it actually enabled us to be able to at least perform experiments without the mechanisms currently being understood about how this is possible. But now, whereas traditionally these arrows and these types of figures have always been drawn in one linear fashion, they actually move in both ways. And so this creates a host of new questions about also where else do these arrows exist and how easily do cells transition between states. But one of the challenges that has still not been addressed by this is that how do we efficiently go from one cell state to any single one of these? Or how do we create the paths by which we direct cells and enlarge populations of these cells to effectively go from state A to B to C in a more defined, reproducible, and robust type of manner? So one of these approaches then, and this is where a lot of interest has been in the field, although not necessarily particularly a tremendous amount of success yet, has been with regards to directed differentiation. This is the idea that we can a priori know what the phenotype is that we're targeting and be able to create the path of the route by which we can get there. If we are effective at that, and that includes, as I depicted here, both the promotion of signaling pathways of different phenotypes or progenitor states, as well as inhibition of others, to really effectively get to homogeneous populations, then the second part is to begin to think about, well, how are we going to do this, as I said, in a way that's large scale enough that it'll actually produce large batches? One of the things from a quality control perspective you wouldn't want is to produce only a few of these multiple times and have to do it in an iterative fashion, because when you get to the quality control stage, you'd essentially have to use all of your product to validate what you had, at which point you now have to start over again. So thinking about how we can do this in a way where we can actually greatly expand upon the success that may be achieved here, and also what are the analytical tools that are necessary to really be able to assess large populations of these cells. They're very, very heterogeneous. There's a lot of noise in these types of systems, and that is a great opportunity, I'll put it as, instead of a great challenge necessarily, for engineering approaches, I think, in particular, to lend themselves to helping with this type of problem. And the last part, then, is in thinking about the application of the cells. It's fairly direct and has been fairly obvious to some extent that if we can produce any single one or different types of these cells, then the direct application for tissue repair processes would be to inject these cells, either the cells alone, cells with scaffold materials, some type of delivery vehicle. What's been interesting is that while that has been largely the experimental and the preclinical approach that has been attempted, and even some limited clinical studies, what typically happens is not that you get cell replacement from those cells that are transplanted, but that actually there's a transient lifetime to those cells, and then after they're gone, there's some alteration that has occurred due to the presence or the introduction of those cells at the time that they were originally introduced into, say, a site of tissue repair or tissue undergoing active remodeling processes. So this creates an alternative or secondary hypothesis, and that is, what else are the cells doing? If they're not just directly replacing the host cells or endogenous cells that may be lost in the case of a disease or injury, then what role are they serving? And if they're serving a role as more of a complex drug delivery vehicle, can we hijack this or jump on this approach and start to think about, instead of using the cells themselves as a therapy, using the molecules that the cells are producing as a potential therapy? So this is largely, and putting into this context, is kind of where the three aspects of where my lab are primarily focused. And these are three areas which are also interrelated with one another, so I try to depict that in this manner here. The first is in how we can begin to try and engineer and control the stem cell microenvironment with the objective of being